Hi everyone, we continue to talk about oxidative phosphorylation. So the Q cycle takes place in complex 3, which is represented in outline form. And in the first half of the cycle, 2 electrons of a bond QH2 are transferred, one to cytochrome C and the other to a bond Q in a second binding site, to form a semiketone radical anion. So the newly formed Q disassociated and enters the Q pool. In the second half of the cycle, a second QH2 also gives up its electrons to complex 3, one to the second molecule of cytochrome C, and the other to reduce semiketone radical anion to QH2. The second electron transfer results in the uptake of 2 protons from the matrix. The path of electron transfer is shown here in red. Cytochrome C oxidase catalyzes the reduction of molecular oxygen to water. The last of the 3 proton pumping assemblies of the respiratory chain as complex 4. Cytochrome C oxidase catalyzes the transfer of electrons from the reduced form of cytochrome C to molecular oxygen, the final acceptor. Boron cytochrome C oxidase is reasonably well understood at the structure level. This enzyme consists of 13 polypeptide chains. Notice that most of the complex, as well as 2 major prosthetic groups, are embedded in the membrane. HemoF3CuB is the site of the reduction of oxygen to water. As molecular oxygen binds, it abstracts an electron from each of the nearby ions in the active center to form a peroxidized bridge between them. 2 more molecules of cytochrome C bind and release electrons that travel to the active center. Reaction between 2 more H1 ions allows the release of 2 molecules of water and resets the enzyme to its initial, so fully oxidized form. 4 protons are taken up from the matrix side to reduce 1 molecule of oxygen to 2 molecules of water. These protons are called chemical protons because they participate in a nearly defined reaction with oxygen. 4 additional pumped protons are transported out of the matrix and released on the cytoplasmic side in the course of the reaction. The pumped protons double the efficiency of free energy storage in the form of a proton gradient for this final step in the electron transport chain. So the figure shows here summarize the flow of electrons from NADH and FADH2 through the respiratory chain. This series of exogenic reactions is coupled to the pumping of protons from the matrix. As we will see shortly, the energy inherent in the proton gradient will be used to synthesize ATP. High-energy electrons in the form of NADH and FADH2 are generated by the citric acid cycle. These electrons flow through the respiratory chain, which powers proton pumping and results in the reduction of oxygen. So look at the table here. Toxic derivatives of molecular oxygen, such as superoxidized radicals, are scavenged by protective enzymes. So superoxidized hydrogen peroxide and species that can be generated from them, such as the hydroxy radical, are collectively referred to as reactive oxygen species, or we call it ROS. Oxidative damage caused by ROS have been implicated in the aging process as well as in a growing list of diseases. What are the cellular defense strategies against oxidative damage by ROS? Chief among them is the enzyme superoxidase dismutase. This enzyme scavenges superoxidized radicals by catalyzing the conversion of two of these radicals into hydrogen peroxide and molecular oxygen. These enzymes perform the dismutation reaction by a similar mechanism. The oxidized form of the enzyme is reduced by superoxidase to form oxygen. The reduced form of the enzyme formed in this reaction then reacts with a second superoxidized ion to form peroxidase, which takes up two protons along the reaction pathway to yield hydrogen peroxide. How are electrons transferred between electron-carrying groups of the respiratory chain? These groups are frequently buried in the interior of a protein, in fixed positions, and are therefore not directly in contact with one another. Electrons can move through space even though a vacuum. However, the rate of electron transfer through space falls off rapidly as the electron donor and electron acceptor move apart from each other. The protein environment provides more efficient pathways for electron conduction. For groups in contact, electron transfer reactions can be quite fast. The case is more complicated when electrons must be transferred between two distinct proteins, such as when cytochrome C accepts electrons from complex 3 or passes them on to complex 4. Cytochrome C is present in organisms having mitochondrial respiratory chains, like plants, animals, and eukaryotic microorganisms. This electron carrier evolved more than 1.5 billion years ago, before the divergence of plants and animals. Its function has been conserved throughout this period. For example, wheat germ cytochrome C reacts with human cytochrome C oxidase. Additionally, some prokaryotic cytochromes, such as cytochrome C2 from the photosynthetic characterism, so we short for RR, and cytochrome C550 from the another bacteria, and they are closely resemble cytochrome C from the tuna heart mitochondria. The resemblance among cytochrome C molecules extends to the level of amino acid sequence. Because of the molecule's small size and ubiquity, the amino acid sequences of cytochrome C from more than 80 widely ranging eukaryotic species have been determined by direct protein sequencing. The striking finding is that 21 of 104 residues have been irradiated for more than 1.5 billion years of evolution. So a polygenic tree constructed from the amino acid sequences of cytochrome C reveals the evolutionary relationship between many animal species. Okay, so here's some information for today, and thank you for your listening. Bye-bye.